Hello everyone and welcome to the 7th student. Today we're looking at C3.1, which is integration of body systems. So this is quite a hard one, but I'm going to try and explain it in the most simple way possible. Let's get started. So, integration. Coordination and integration is really important to perform all of the functions in life. So there's a lot of levels to integration, right? So for example, we have organelles integrate to form cells. Cells integrate to form a tissue, such as cardiac tissue. Tissues integrate to form an organ, such as the heart, right? And then organs integrate to form a system, such as the circulatory system. Systems then integrate to form an organism. So, because in multicellular organisms there's specialization to perform specific functions, integration is needed to carry out functions at large enough scale. And this leads to what's called emergent properties. So, for example, the cheetah is an effective predator, but that is only because of the integration of its systems, which at the same time depend on integration from its organs and so on and so on. You get the gist. For example, having the muscular system is key to its speed, right? But the muscular system also depends on the circulatory system to provide fresh blood to the muscles, and the gas exchange system to provide oxygen to the blood. So I think you get the gist, right? It's really important that there's integration in multicellular organisms. So this topic is a bit random in my opinion because it has a lot of different things, but I've tried to organize as best as I can. And I think the three main pillars of this topic are that you have three requirements for integration, right? So first, you need communication between parts. So that's performed by the endocrine and nervous system, which we looked at in the past two topics. You need to remember the speed and duration of the responses and the destination types of signals. So check out the previous videos if you do need a recap on that. I'm not going to go over it again. Then we also need an information integration system, right? And that's done by the central nervous system in humans. So the brain and the spinal cord. The brain receives information from sense organs, such as the eye, and then can store that information. So that's what short-term and long-term memory is, right? That's essential for learning. So this information is then processed, and that leads to decision-making through sending signals to one area or another. The spinal cord, for those of you who don't know, is found in the backbone, and it's made of white matter and gray matter. And it only coordinates unconscious processes, specifically the gray matter, such as reflexes. We'll look at those in a sec. Unconscious processes are involuntary, so we cannot prevent them and we don't have to think about them to do it, right? Such as contracting the heart. We don't think about that. And then the third thing that we need is a transport system. So in our case, the circulatory system, right? This serves all organs by bringing them oxygen and glucose, as well as water, carbon compounds, and removing waste products, right? So something to bring substrates in and take the waste away. Okay, let's look at an example of how this works in real life. So, for example, when we need to respond to changes in the external environment, we perceive that through sensory receptors. So some sensory neurons, okay, and this is where the terms start to get confusing. So some sensory neurons have nerve endings which can detect stimuli, for example, heat. But other stimuli, okay, require specialized receptor cells. And then those pass that onto the sensory neuron, okay? So it can start with the receptor cell or the sensory neuron, important distinction. Regardless though, sensory neurons then convey the messages to the CNS, the central nervous system. So the cerebral hemispheres of the brain, specifically the primary motor cortex, okay, that's an important word to remember, then send the signals via motor neurons to striated muscles in the body, causing them to contract and thus movement. Okay. Also important to know, both sensory and motor neurons are normally found in nerves. Nerves are a bundle of nerve fibers encased in a protective sheath. However, some nerves might only contain sensory neurons and some might only contain motor neurons. Okay, so nerves don't necessarily contain both. Okay, so now we're going to look at the reflex arc. But before we do so, I want to introduce my new website where you can get all the drawings in IB Biology as slides in case you want to have it to label or draw yourself, and also all of the slides that I use in these topics in case it's useful to annotate alongside when you watch the videos. So if you want to check those out, they're at the7student.com. I'll link that in the description in the comments, and you can always take a look later. But moving on. So let's look at reflex arcs. How do these work? Okay, so these have to be really quick, right? Because we need to respond to things like pain or heat. So they involve a very small number of neurons to make sure that it is fast. And importantly, it is rapid and involuntary. Those are key terms, okay? So as we talked about, the receptors or sensory neurons directly detect the stimulus, For pain or heat, it's normally the nerve endings of sensory neurons, and this is sent to the CNS through neurons with really, really long axons. So those go all the way to the spinal cord or the brain. The brain mainly gets signals from the main sense organs found in the head. The spinal cord mainly gets signals from other organs in the body, including the skin and muscles. So these axons then end at synapses with interneurons, okay, or relay neurons. This is found in the gray matter of the spinal cord or the brain. And then interneurons or relay neurons have a lot of dendrites and process signals by combining impulses from multiple inputs. Basically, the relay then sends it to the motor neuron, okay, that's the most important part, which the motor neurons then receive the signals via synapses and relay it to the effector. So the effector can be a muscle, which can contract to move, or glands, which secrete hormones. Main idea is sensory neuron to relay neuron to motor neuron, okay? Cool. So now, this is where it starts to get a bit random with this topic, okay? There's just a lot of random concepts thrown about, but the cerebellum. So the cerebellum, okay, you need to know, coordinates and fine-tunes skeletal muscle contractions as well as balance. It allows very, very precise coordination of movements and helps us do activities requiring motor memory, such as riding a bike or typing a keyboard. That's why people who fall on the back of their necks often damage their cerebellum and they forget how to do these things that we take for granted. Good. So that's it for the cerebellum. Okay, just remember those key things on screen. Next, circadian rhythm. So humans are adapted to live in a 24-hour cycle, and we follow circadian rhythms, which are controlled by the hypothalamus, specifically the suprachiasmatic nuclei, okay, or SCN, in the hypothalamus. So this controls the secretion of melatonin by the pineal gland. And this increases melatonin in the evening and then drops to a low level at dawn. So high melatonin is what causes drowsiness and promotes sleep. So this makes sense, right? Then during sleep, the liver removes melatonin from the blood, and that encourages waking up. So again, this is something else to remember. Remember this pathway. I know it's a pain to remember all of these pathways, but you really do need to know them. Okay. Now we're going to look at epinephrine. So epinephrine or adrenaline, it's the same thing, okay? It's a hormone that prepares the body for vigorous physical activity. So it's the fight or flight response, okay? So it's controlled by the brain, by the amygdala, and that sends distress signals to the hypothalamus, which then sends signals to the adrenal gland, okay, causing adrenaline release. And what it does is it basically increases the supply of glucose and oxygen through increasing glycogen breakdown and increasing the ventilation rate. And also it causes the sinoatrial node to speed up the heart rate and arteries, which go to muscles and liver, to widen so that more blood flows to them, basically in preparation for, again, physical activity, right? We'll look in a second at how that's controlled, specifically the sinoatrial node part. So keep this in mind. But before we get to that, though, we need to talk about the hypothalamus. So the hypothalamus is really, really important, okay? It acts as the link between the nervous system and the endocrine system. So it's a small region in the brain, okay? And it has a specialized area called nuclei. So nuclei receive signals from sense organs, but they also have sensors themselves. So it's receiving a lot of information, and it interacts a lot with the pituitary, which is a gland located right below it. It has an anterior lobe and a posterior lobe. And an example of this is osmoreceptors. So osmoreceptors in the hypothalamus measure the solute concentration of blood, and that influences how much ADH is secreted. Similarly, with puberty, it's through secreting GnRH, which then stimulates secretion of LH and FSH by the pituitary, okay? So just know that this controls, it's kind of the master regulator for a lot of things, this pathway. We're going to look at an example right now. So this is what I was talking about, okay? This diagram is really complex. I know it's really difficult for a lot of you guys, so let's go through it slowly, okay? This is how heart rate is controlled. It's important to note that this is the centerpiece of the diagram, okay? The sinoatrial node is the part of the heart, it's found in the right atrium, that controls the heart rate, okay? That's why it's called the pacemaker, because it initiates the signal and causes the heart to contract. So this is the first pathway through which you can activate heart rate, okay? This arrow should be pointing the other way. Apologies. So this is what we just talked about. The amygdala, when it detects fear or opportunity, can stimulate the hypothalamus to activate the adrenal gland. And then that stimulates epinephrine release, which activates the sinoatrial node to speed up the heart rate, okay? So that's one pathway. That's when there's, again, fear or excitement. But then the normal pathway through which heart rate is controlled is this one over here, whereby you have these receptors in your artery walls, okay? So baroreceptors measure pressure. So if pressure is too high, what's going to happen? Well, if it's too high, it's going to try to reduce the heart rate, right? So that it decreases. And then the chemoreceptors detect blood pH, meaning CO2 levels, okay, because high CO2 makes the blood pH lower, and also oxygenation. So if there's too little oxygen and too much CO2, again, you're going to want to increase the heart rate. These work through negative feedback. Okay, it's important to remember, this is a common theme. Negative feedback always returns values to baseline. So it's used a lot in homeostasis and regulation in the body, okay, because we don't want to do self-amplifying loops. We want things to return to normal. Basically, what that means is that if I detect high pressure and I reduce the heart rate, right, then what's going to happen is the pressure is going to decrease, right, because the heart rate has decreased, and then that's going to do negative feedback, right? So then these receptors are going to inhibit the heart rate from increasing and are going to actually downregulate that, right? And similar with the chemoreceptors. If they detect too much CO2, then that's going to increase the heart rate, which is then going to make sure that there's less CO2, and then that's going to inhibit the heart rate from increasing even more, okay? They do so through the cardiovascular center, which is found in the medulla. So the medulla is found here in the brainstem, and then that triggers the sinoatrial node through the vagus nerve and the sympathetic nerve. The sympathetic nerve increases the heart rate, and the vagus nerve decreases it, okay? Next thing, which is also intertwined with that, okay? And it's how do we control ventilation rate? Well, in a similar manner. So basically the same chemoreceptors, if they detect a low pH, meaning a high CO2 concentration, they're going to activate the respiratory center, found again in the brainstem, not the medulla, but the brainstem. And that causes the diaphragm and external intercostal muscles to contract. When these contract, you inhale, right? So that controls ventilation rate. And then the lungs expand. But this is also negative feedback, because when the lungs expand, okay, stretch receptors detect this and they inhibit the respiratory center from sending signals, meaning you decrease your ventilation rate, okay? So once again, it's about getting it back to baseline. This is the last thing for those of you in SL, and I have to say this is probably one of the most complicated topics in SL, but in HL there's not much more. So let's finish this off. So last thing, okay, is that is gut movements. So there are only 2 gut movements that are voluntary and thus under control of the central nervous system, and that is swallowing food until it reaches the esophagus and defecation, okay? Everything else is under the control of the enteric nervous system. So peristaltic muscle contractions, okay, are controlled unconsciously. Peristalsis is a very key word, okay? It's what makes sure that the food goes down your whole gastrointestinal tract. So how do these work? Okay, there are waves of vigorous contraction. So the walls of the gut basically have 2 layers of muscle tissue in opposite orientation. So you have smooth circular muscle, which contracts behind the food to ensure it's not pushed back. And you also have smooth longitudinal muscle, which moves it along the gut, okay? So the smooth muscle can exert continuous moderate force, and then also short periods of more vigorous contraction is done through the striated muscle, okay? So that way you ensure the food goes down, but also doesn't go back up. Great. So now, for those of us in HL, let's keep going for a little bit. Tropic responses in plants. This is easy. So plants control the direction of growth of their roots and shoots through responses to directional stimuli called tropisms. So most roots are gravitropic, meaning they grow downwards in the same direction as gravity. And then most shoots are positively phototropic and negatively gravitropic, meaning they grow towards the source of light and in the opposite direction to gravity. So to grow towards the brightest light, you need to respond by differential growth, meaning one side of the plant needs to grow more than the other one, causing it to curve. What happens is the shadier side grows at a faster rate, so then it flops, right? It kind of curves towards the direction of maximum light intensity. I know it's a bit counterintuitive for a lot of you guys, but it doesn't make sense if you think about it, right? If one side grows larger, right, then the other one automatically bends. And then once it's already looking in the direction of the sun, both sides will grow equally fast. So how is this controlled? Well, we have phytohormones. So phytohormones help to control growth, development, and responses to stimuli. So they can promote or inhibit growth by affecting cell division and cell enlargement. That's the first thing, growth, such as, for example, auxins. They can also promote or inhibit development. So, for example, ethylene does that, right? It can promote or inhibit fruit ripening. That's a type of development. And then responses to stimuli, such as tropic responses, are also controlled by phytohormones. So, jasmonic acid is an example of this. It can trigger the secretion of enzymes to digest a fly in flytrap plants. That's an example of response to a stimulus. Okay. So, auxin efflux carriers. This is maybe the hardest thing in this topic. So how do auxins promote cell growth? Okay, first we need to understand how are auxins actually causing the cell to grow. Okay, you need to understand a plant cell has these cell walls, right? So those are quite rigid. You need to break them to make the cell grow. So what auxins do is they promote, and this is hard, okay, but so listen up. They promote the synthesis of proton pumps, okay? So then these proton pumps are inserted into the plasma membrane, so inside here, right? Then these transport protons to the cell wall, okay? Clear thus far. So these protons then are in the cell wall, so they're acidifying it. And what this does is that the decrease in pH weakens the cell wall, causing it to break and extend, right? So the cell can elongate that way. So that's how auxins actually work. But then how do auxins move between cells? So it's, again, quite complicated. Auxin can enter cells by passive diffusion, but once it's inside, it can't leave, okay, because of chemical changes. So the only way for it to leave is through carriers, so auxin efflux carriers. These can pump it into the adjacent cell wall, where it can then diffuse into the next cell over. And basically, by controlling the distribution of these efflux carriers, you generate concentration gradients, and you can control where your auxins are, and thus what parts of the plant grow, okay? So hopefully this diagram and my explanation are somewhat useful, but if there's questions on this, do leave them in the comments, because I understand that it's quite hard to understand. Okay, this is super easy. You just need to know that basically auxin interacts with cytokinins. So cytokinins are another type of hormones. And so whereas auxin is produced in shoot tips and then transported down shoots, cytokinin is produced in root tips and then transported up. So amounts of each balance out root and shoot growth, right? So sometimes they work together to simulate a process. That's synergism. Sometimes they have opposing effects. That's antagonism. So for example, hedges, right, such as this one, need cutting off shoot tips. So removing sources of auxin, because auxin inhibits side shoot growth, and that allows them to grow, right? So if you remove auxin, you make sure that they grow to the sides and not only up. Cytokinin stimulates side shoot growth. So if you remove the auxin, only the cytokinin is present and they will grow to the sides and not just up, which is what you want with hedges. Okay. And then finally, okay, last thing is that fruit needs to ripen because when the seed is fully developed, you want animals to eat it so that the seed will be transported. And in a lot of species, this is stimulated by ethylene. So ethylene, we mentioned it before, it's a phytohormone. And this works through a positive feedback mechanism. So basically, ethylene causes ripening, and then ripening fruits produce ethylene. So self-amplifying, right? And then ethylene is released as a vapor, right? Because it's volatile. So it helps synchronize ripening of fruits on a plant. Because once one starts to ripen, it'll release ethylene into the air and it'll go into other fruits. Okie dokie. So let's do some questions. What's the primary function of negative feedback mechanisms in the body? I'll give you some time to think. You can pause now and think about it, but I'm going to count down in 3, 2, and 1. Homeostasis. As I said, homeostasis is all about returning things to baseline, right? To a base level, such as 37 degrees for temperature, for example. So negative feedback is perfect for this because it's not self-amplifying, right? It controls itself. Whereas if you want to amplify a physiological response, then that would be positive feedback, right? And probably same thing for hormonal secretion or cell division. Okay, great. Next question is, which of the following describes the sequence of components in a simple reflex arc? This should be pretty straightforward, I hope. So 3, 2, and 1. B. Okay, so you have a receptor that goes to a sensory neuron, that goes to the relay interneuron, to the motor neuron, to the effector. Okay, obviously it's not the effector first. It's not the muscle that detects something. The issue with C, okay, is that the effector is not before the motor neuron. The motor neuron tells the effector to do something. And then D is obviously wrong in a lot of ways, right? But the motor neuron goes at the end. It's the one that tells the effector what to do. So it doesn't detect anything itself. And last but not least, what role does the medulla oblongata play in the regulation of heart rate? Okay, again, pause now. And in 3, 2, and 1. Okay, so it adjusts heart rate in response to changes in blood CO2 concentration through the chemoreceptors, which detect pH, right? So it does not generate electrical signals to initiate heart contractions. Heart contractions are already happening, right? It just speeds it up or slows it down. It does not secrete adrenaline. That's the adrenal gland, remember? And it does not block sensory inputs in any way from affecting cardiac function, right? In fact, it's the opposite. It detects the sensory inputs from the baroreceptors and the chemoreceptors, and then it affects cardiac rates. Okay, amazing. So I know this was a long one and quite a tough one. So any questions, leave it in the comments. And otherwise, I'll see you next week for the next topic. Bye, everyone.